Welcome to today's lecture on the topic of pharmacology. In this session, we will delve into the fascinating world of cancer chemotherapy and discuss various classes of drugs that are commonly used in the treatment of cancer. Cancer is a complex disease characterized by uncontrolled cell growth and the ability to invade other tissues in the body. Chemotherapy drugs act by targeting different aspects of cancer cell growth and division. Alkylating agents are a class of chemotherapy drugs that directly damage the DNA of cancer cells. These drugs work by forming covalent bonds with the DNA molecules, leading to the formation of crosslinks between DNA strands. This disruption in the DNA structure prevents cancer cells from dividing and ultimately leads to their death. Examples of alkylating agents include cyclophosphamide, cisplatin, and busulfan. These drugs are frequently used in the treatment of various types of cancers, including lymphomas, leukemias, and solid tumors. Antimetabolites are another class of chemotherapy drugs that interfere with the normal metabolic processes of cancer cells. These drugs mimic the structural components of DNA, RNA, or proteins, and when incorporated into cancer cells, they disrupt their normal functioning. Antimetabolites can inhibit DNA synthesis, RNA transcription, or protein synthesis, ultimately halting cancer cell growth. Common examples of antimetabolites include methotrexate, 5-fluorouracil, and gemcitabine. These drugs are frequently used in the treatment of breast, lung, and colon cancers. Platinum-based drugs are a class of chemotherapy agents that contain a platinum atom as their central component. These drugs act by binding to DNA and forming adducts with the DNA molecules, leading to cross-linking and DNA damage. Platinum-based drugs, such as cisplatin and carboplatin, are widely used in the treatment of various solid tumors, including ovarian, testicular, and lung cancers. Topoisomerase inhibitors are chemotherapy drugs that interfere with the action of enzymes called topoisomerases. These enzymes play a crucial role in the manipulation of DNA structure during replication and transcription. By inhibiting topoisomerases, these drugs disrupt DNA repair mechanisms in cancer cells, leading to the accumulation of DNA breaks and eventual cell death. Examples of topoisomerase inhibitors include etoposide, irinotecan, and topotecan. These drugs are commonly used in the treatment of leukemia, lymphoma, and lung cancers. Microtubules are essential structures involved in cell division and intracellular transport. Microtubule targeting agents are chemotherapy drugs that interfere with the function of microtubules and disrupt cancer cell division. These drugs can stabilize microtubules or inhibit their assembly, leading to cell cycle arrest and cancer cell death. Examples of microtubule targeting agents include paclitaxel, vinblastine, and vincristine. These drugs are used in the treatment of breast, lung, and ovarian cancers. Histone deacetylase, HDAC, inhibitors are a class of drugs that interfere with the activity of DAC enzymes, which play a role in the regulation of gene expression. DAC inhibitors can promote the acetylation of histones, leading to changes in chromatin structure and gene transcription. These drugs have shown promise in the treatment of certain types of lymphomas and leukemias, such as cutaneous T-cell lymphoma and peripheral T-cell lymphoma. Examples of DAC inhibitors include vorinostat, romidepsin, and belinostat. Poly-ADP ribose, polymerase, PARP, inhibitors are a relatively new class of drugs that selectively target cancer cells with defects in DNA repair mechanisms, such as those with mutations in the BRCA genes. These inhibitors block the function of PARP enzymes, which are crucial for DNA repair. By inhibiting PARP, these drugs prevent cancer cells from repairing DNA damage and induce synthetic lethality. PARP inhibitors, such as olaparib, rucaparib, and niraparib, have shown great promise in the treatment of ovarian and breast cancers. Proteasome inhibitors are drugs that interfere with the activity of proteasomes, cellular complexes responsible for the degradation of proteins involved in cell cycle regulation, DNA repair, and apoptosis. By inhibiting proteasomes, these drugs disrupt the normal protein turnover in cancer cells, leading to cellular stress and apoptosis. Bortezomib, carfilzomib, and ixazomib are examples of proteasome inhibitors that are primarily used in the treatment of multiple myeloma. Epidermal growth factor receptor, EGFR, inhibitors are targeted therapy drugs that specifically target cancer cells that overexpress EGFR, a receptor frequently found on the surface of various cancer cells. These inhibitors can block the signaling pathways involved in cell growth and survival, thereby inhibiting cancer cell proliferation. Examples of EGFR inhibitors include erlotinib, gefitinib, and cetuximab. These drugs have shown efficacy in the treatment of lung, colorectal, and head and neck cancers. The mammalian target of rapamycin, mTOR, is a protein kinase that plays a crucial role in cell growth and metabolism. mTOR inhibitors are drugs that block the activity of mTOR, inhibiting cancer cell growth and survival. Drugs like everolimus and temsirolimus are examples of mTOR inhibitors that are used in the treatment of renal cell carcinoma, breast cancer, and pancreatic neuroendocrine tumors. Immunomodulatory drugs, IMiDs, are a class of agents that modulate the immune system to enhance anti-tumor responses. IMiDs, such as lenalidomide and pomalidomide, have immunomodulatory, anti-angiogenic, and direct anti-tumor effects. These drugs are commonly used in the treatment of multiple myeloma and certain lymphomas. Chimeric antigen receptor, CAR, T-cell therapy is an innovative and personalized approach to cancer treatment. This therapy involves genetically modifying patients' own T-cells to express CARs, which are receptors capable of recognizing specific antigens on cancer cells. By redirecting T-cells to target cancer cells, CAR T-cell therapy has shown remarkable clinical outcomes in hematological malignancies like acute lymphoblastic leukemia and non-Hodgkin lymphoma. In conclusion, cancer chemotherapy is a vast field that encompasses numerous classes of drugs targeting different aspects of cancer cell growth and survival. The diverse mechanisms of action of these drugs provide oncologists with a wide array of treatment options for various types of cancers. Understanding the pharmacology of these drugs is essential for medical professionals involved in cancer treatment and research.